Okay, so on to the muscular system review. So we've got a couple things that we want to go over here. We're going to break this up into two parts. Just figured I'd have two short videos as opposed to one long video. So we've got proteins that make up what's called the myofibril. All right, we've got myosin. That creates movement by moving actin. So let me turn on my pen here. So myosin is this purple here, right here. We've got actin, which is the green here. Okay, so we've got troponin, which we see down here, and it's these little globules here. Okay, and what troponin does is it moves tropomyosin out of the way, and tropomyosin are these long orange strands. Okay, and they block. They prevent actin and myosin from getting together. Then we've got titin, and we're going to talk more about titin later. It's actually pretty interesting. It's an elastic protein. Okay, and so we've got titin up here. Okay, it looks like a spring. And what that does is it helps stabilize actin and myosin and actually might help generate more pressure. We'll get into, or more force, excuse me. We'll get into that later. But it's one of its main jobs or functions appears to be that it returns the muscle to resting length. And then we've got nebulin. And so nebulin, and I'm not sure if it's on here. I'm not seeing it right now on here, but it's an inelastic protein that's kind of interwoven with some of these. All right, but we're not going to focus a whole lot on it. But one of the things I want you to realize is that they come in pairs. Okay, myosin and actin, troponin and tropomyosin, and then titin and nebulin kind of go together. Like it says, stabilizes actin. Now within a sarcomere, we've got kind of an anatomy, or we definitely have an anatomy. It goes from Z line to Z line. Okay. Now within a myofibril, there's many sarcomeres. Now we can break this up into a couple of different things. So we've got the I band, the A band, and the H zone. Okay. Now notice that in the micrograph here, the I band is white. Light gets through it. That means isotropic. The A band, on the other hand, light doesn't get through as easily, and it's called anisotropic. And then we've got this H zone, which looks a little bit different. Okay. And so just to show you here, the A band is wherever we have actin. A is for actin, or excuse me, myosin. So A is for, I think of it all of the myosin, excuse me. So I probably messed that up. So A is for anisotropic, and that's where all the myosin is. I band is where we just have actin, no myosin. That's the light band. And then we've got this H zone right here, and that's where we only have myosin. Okay. Now notice as we contract the muscle, the A band stays constant, but the I band and the H zone get smaller. Okay. Now the last thing that I want to do is go through the neuromuscular junction here. Okay. And so we're going to go through this pretty briefly here. So neuromuscular junction is made up of just what it sounds like. We've got a neuron and we've got a muscle cell. Okay, action potential comes down, ultimately causing the release of acetylcholine. Acetylcholine will bind to these blue things, which are ligand-gated sodium channels. That causes or allows sodium to come in. That will depolarize the cell and open up some voltage-gated sodium channels. And notice we've got sodium channels that go all along. And basically what happens is an action potential goes all the way down here into the T-tubule. And once it gets there into the T-tubule, it activates this DHP receptor, okay, which is a voltage-gated calcium channel. Okay, the voltage-gated calcium channel also has a string attached to it. And it opens up the ryanodine receptor, or activates it. That ryanodine receptor, which is this green door here, is a calcium channel. It opens the door and calcium will flood out, ultimately allowing actin and myosin to bind together. Okay, remember we need calcium in order for actin and myosin to bind together. And so that's where we're going to end for this one.